Welcome to our the first seminar of the season. We're really appreciative to all of you for braving the snow. We're even more appreciative of our speaker who left sunny North Carolina to come and speak to us. And there's quite an anecdote to retell about Dr. Steve Kritchevsky's visits to the HNRCA. The last time Dr. Kritchevsky came to give the HNRCA seminar, it was a tough snow day. So this is 2 out of 2, and he actually stayed overnight at Dr. Joseph Cahias' house, and he gave the seminar after that on the 8th floor conference room. And there were so many people who came the next day. We have fond memories, Irv, of you inviting a certain female scientist to sit on your lap because there was no other room. So, we are just very honored to have for our first seminar this season, Dr. Stephen Kritchevsky. For many of us, he needs no introduction. But for those of you who are not familiar with Steve, he is actually the director of the J. Paul Stick Center on Aging at Wake Forest in Winston-Salem, North Carolina. He's also the vice section chief of the section of gerontology and geriatric medicine. He's the associate dean for research development. He's their associate director of the Translational Institute. He has a very hefty CV, which we won't go through because I think we should leave the time for what's going to be a really fascinating talk on the biomarkers of human longevity. I just want to point out that any trainee who would like to meet with Dr. Kritchevsky after the talk, we are having lunch on the 9th floor, and Dr. Kritchevsky actually is the PI of training grants for postdocs, so interested people should go to the lunch. So without further ado, I'd like to introduce our speaker and the talk for today. Thank you. If you want do you want this, or are you fine?
They told me this would work, and
okay.
I'll just hang out up here. Thank you very much, Sarah. Delighted to be here. I'll have many friends here. And so I'm just sorry for the weather I brought. I just want you all to know it's always like this in North Carolina. It's always sunny. It was 54 degrees when I left. And we've heard of snow, haven't seen much of it, but we know it exists. So I would invite any of you who'd like to come and visit, especially those interested in postdoctoral fellowships, have a really great program for that. So I'm going to talk to you a little bit. The title of this talk is called Biomarkers of Human Longevity. And I want to tell you how I came to this came to be thinking about this. I've had some interactions with people in biology of aging. In biology of aging, people feel like they've almost got aging cracked biologically. Over the last 15 years, 20 years, the number of metabolic pathways that are evidently related to how long animals live in controlled experimental environments has revealed all kinds of interesting pathways. And some of which are druggable targets. And so everyone's very interested in translating these biologic findings from animal models into human models. And the question is, how would you do that? What would be a strategy for doing that? And I'm going to argue that to do this, the only feasible way is to have some kind of markers about what these interventions would be likely to do over the long term, and talk about some candidate biomarkers that might be considered. I'm not going to claim that this is the list, but I think thinking about it helps to think about what good candidate biomarkers might look like. And then how would you validate those biomarkers? And I'm going to argue that since most of the metabolic pathways that have been identified in the biology of aging have been revealed through caloric restriction experiments, maybe we can use weight loss trials in humans, and I'd particularly argue older humans, as a way of validating candidate biomarkers for use when evaluating other kinds of interventions. So everyone has seen something like this. If you're in an aging center, you have a model where either the whole person or a particular organ system declines in some of its capabilities through time. After a while, this begins to be reflected in the loss of ability to do certain things. When this gets really bad, you can't do those things on your own. It's called disability. And when it gets really, really bad, it's incompatible with life and you die. So, you know, people have presented this and many more complicated, nuanced versions of this. But basically, this suggests really that there are two ways to prevent disability and extend life. And one is you add reserve. So you take the whole line and push it up, and then it just takes longer for you to get to these thresholds. And this actually supports a life course view of aging. If you want to prevent fracture, for example, a good time to intervene is in late teens and early 20s when most of your own bank account is deposited in. Is that? I sometimes lose myself in my own metaphors, so if I do that, excuse me. We know from work like Dr. Fielding does, I don't know if he's in the audience today, that muscle function, you can add reserve no matter how old you are. You can exercise people in their 90s and they can get stronger and have functional benefit. The people in the biology of aging would rather affect this line. They want to slow the rate of decline. The hypothesis is there's some fundamental underlying processes of aging that if you can decelerate them, you will gain lifespan and gain active life expectancy by slowing the process of aging. So there are now treatments to slow aging-related decline. They're here now. If you're a mouse, these have been shown in mouse models. I think this is probably, if you look at the science from the rodent perspective, there's never been a better time to be a rat or a mouse. We know how to prevent heart disease in you. We know how to prevent cancer. We can make you live longer. They still die. Yeah, I know. So I think notable in this explosion of knowledge, which I've referred to, is a testing regimen that the National Institute of Aging is supporting called the Interventions Testing Program, which is a very structured way of evaluating candidate life-extending molecules in mouse models. And they have identified 4 treatments so far that they feel will expand lifespan and probably health span in rodent models. These are metformin, rapamycin, acarbose, and I believe it's alpha estradiol is the fourth. Now, not all of these work in both sexes of mouse. Sometimes only one does better than the other, but they believe this is a paradigm for the early evaluation of treatments that have the potential to perhaps extend life in humans. However, the evaluative framework they're using in the mouse model really can't be applied to humans. So what they do is they start giving animals these interventions either at 4 months or 12 months. So if you do sort of the mapping to human lifespan, that's the same as starting at 18 years old or about 40 years old. So I can tell you what's not going to happen. We're not going to give some new compound to 10,000 18-year-olds and wait 100 years to see who dies and who doesn't die. I can tell you something else we're not going to do. We're not going to give 5,000 or 6,000 40-year-olds a drug that may have no benefit, may have side effects, and give it to them for 50 or 60 years to see who lives longer, who doesn't. So we're at a sort of translational crisis of how you take this biologic information and go on to begin to evaluate whether or not there's promise in human studies. So I think there are several strategies that can be pursued for evaluating whether these treatments will have the expected effect in humans. The first, I think, is fairly, I guess none of it is completely straightforward, but is to observe anticipated physiologic and molecular responses in relevant tissues. So if you put animals on caloric restriction, mTOR goes down, various pathways go up, inflammation goes down. These are a whole set of pattern responses that happen in relatively short order that are observable in analyses of muscle biopsies or other biopsies from other tissue. And so you could certainly do this to see if it's having the expected physiologic effect at the cellular level. The problem is, I think, from my perspective, is the connection of those molecular changes with disability and longevity phenotypes, at least to me, is not entirely clear. It's sort of, okay, we made these changes, physiologic changes, yada, 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 that mice die. There's a whole lot of stuff going on between that and the end, and some of it is reflected in slower rates of loss of kidney function or cardiac function, but really it's we'd like to know a lot more about what happens between the cellular changes and the mortality phenotype. Another strategy is to observe an improvement in an intermediate endpoint or biomarker that's related to mortality and see if it goes in the expected direction. An example might be muscle function. One of the hallmarks of disability is weakness. It also greatly predicts mortality in older people. If you gave them something and they became stronger, that would be a very interesting thing to avoid. To do that evaluation, you need to avoid ceiling and floor effects. So if you're looking at a muscular readout or a vascular readout, you wouldn't do this in marathon runners because you're unlikely, no matter what it did, see it in someone so advanced. Or you don't want to do it in people who are so ceiling or floor, depending on which way it goes. Older populations are attractive from this standpoint because there's lots of heterogeneity. You get lots of people who are not at the top of their game. They have room to go up, room to go down. These kinds of studies tend to require small to moderate sample sizes, and it really depends on the duration of the study, depends on what biomarker you're looking at and the plasticity of the target tissue. Muscle is plastic. You can regenerate it, you can grow it at almost, to some extent, almost at any age. Kidney tissue is not plastic. You can't get more kidney function by intervening on the kidney. So that leads us to the next strategy, which is to observe the slowing of change over time in a biomarker intermediate endpoint. So here you need a population with enough anticipated change to observe a differential effect. So you wouldn't want to do this in 25-year-olds and follow them to 35 because people are pretty much at a plateau of many physiologic functions and they're not going to change anyway, so anything you give them, you're unlikely to see any benefit or lack of benefit. But in older people, there is expected change and expected acceleration in many organ systems that you could be used to evaluate as an intermediate endpoint on the way to mortality. Things like kidney function, for example, goes down. Lung function, FEV1 is an example. Hard to see how that goes up, but it certainly goes down, and lots of trials in both kidney and lung are focused on not improving function, but slowing the rate of decline of function of those systems. These take longer to do and require a longer sample size because it's hard to know in advance what the variability is going to be in often many years, so the change you observe is greater than the natural intra-individual variability that makes it hard to see change. And finally, we could look for differences in life expectancy and mortality, but to make this feasible, you would need a high event rate or a very large sample size or a very long duration. And usually some combination of the three. And to make it feasible to look at mortality, you might want to do this in older people. So the point I'm trying to make through all of this is that early evaluative paradigm for some of these life-extending treatments probably will work best in people who are older rather than younger, because you have the variability, you have the expected change over time, and something you can sort of get your hooks into statistically and analytically to be able to demonstrate a benefit. So I thought and continue to think that weight loss trials in older adults can guide the design of longevity treatment evaluation. And I've already sort of laid out that argument. Much of our current understanding of longevity pathways are based on caloric restriction models. The short-term caloric deficits achieved in human trials are often of a magnitude that is associated with life extension in animal models. And so when evaluating interventions that are targeting the same pathways that weight loss targets, it makes sense that we might use weight loss trials to provide a starting point regarding what biomarkers are responsive to the intervention and what are plausible effect sizes. So I'm coming from this from an epidemiologic statistical tradition, and we're always thinking about how big is the effect, because that tells us how long the study has to be and how many people we have to study. So this is an example of that first strategy, observing anticipated molecular responses in humans. This is an interesting study by Merkin and others that came out a few years ago. There's a group of people that Luigi Fontana has corralled who have, on their own volition, have voluntarily calorically restricted themselves. So they eat on purpose less than, and there must be miserable people, but they're hungry all the time. But nonetheless, they're very thin, thin people, and they're just trying not to eat as much people. And they compared their gene expression patterns to people of comparable age, also non-obese, who were not voluntary caloric restrictors. And this is a summary slide of all the things that they're seeing. And it turns out this replicates very much everything you'd expect to see from a caloric restriction model in animals. You get a suppression of inflammation, sirtuins go up, mTOR goes down, antioxidant responses go up, there's mitochondrial biogenesis, higher mitochondrial content. So all these changes that are seen in animal models of caloric restriction are recapitulated cross-sectionally in humans who do this on their own. Whether these people will live longer, we don't know. They may not live a long time, but to them it'll feel like it's a long time. So this is a completely reasonable approach to take. But for human older people, we are interested in these other physiologic biomarkers, and so we can ask the question, are longevity-associated biomarkers responsive to intentional weight loss? So if we could do it directly, this is what we would do. We'd have an intervention and wait and see if someone died or not and randomize this, and then if there was a difference, then we'd know what the answer was. But for the reasons I've already elucidated, we can't do that. We can use observational epidemiologic research to link differences between people in markers and their risk of death. And we can do intervention trials in similar people and look at change in biomarkers. But the drama here is we don't know that the change in biomarkers to achieve differences will give you the same effect on mortality or not. This is a hope, a guess. We don't know. So for some things, FDA has already accepted things as biomarkers for drug development. Hypertension, blood pressure is an accepted biomarker for stroke and cardiovascular disease. You don't have to, if you have a new blood pressure medication, you don't have to prove it prevents stroke. All you have to do is prove it lowers blood pressure as well as existing things, and you can get that license. Cholesterol for heart disease is another biomarker that exists. So what is a biomarker? Well, we could have age-related biologic changes that lead to a change in a marker level that directly cause death or disease. This is the hypertension, blood pressure, stroke model, cardiovascular disease, cholesterol, heart disease model. And this is ideal. The trouble is we don't know exactly what causes people to die in every case. So the marker level, the thing that's leading directly to death and disease in many cases is a black box to us. But what we can do is observe other changes associated with aging that may be a proxy for something we can observe, almost like a latent variable of your, I guess, biologic age to some extent. And see, and these are also related to death and disease, maybe causally, maybe not. So what biomarkers? What would be good stand-ins? So these are a short list of potential criteria that you might use for selecting biomarkers. You'd want it to change with chronologic age. If you say it's a stand-in for age, it better be correlated with age. You'd want to see mortality prediction robustly across many, many studies in populations, different comorbidity groups, just very pervasive prediction. So it's not reflecting a particular disease entity, but it's reflecting something more global. This is not necessary, but nice to have, is a monotonic increase in mortality rate across a range of the biomarker. A lot of biomarkers in aging, physiologic measures, have a U-shaped relationship with mortality. People on the very low side or the very high side have excess mortality, and the people in the middle. But if I'm doing an experiment and it goes down, I don't want to get confused that maybe it's too low and going up the other horn or something like that. It's nice if it's measured and not self-reported. That helps standardize things across cultures and clinics and that sort of thing. There are some very good self-reported things that satisfy these criteria that are self-reported. Self-reported health, for example. How good is your health? Good, fair, excellent, poor. No matter what the situation, people who say their health is fair or poor, they're right, and they're much more likely to die in any time span that I've seen than people who self-rate their health as good and excellent, regardless of whatever diagnoses that you might control for, by the way. And the mortality prediction is independent of age, also a good criteria. So I've been involved in the epidemiology of aging for about 20 years. I've got a chance to see a lot of different data sets, and I've come up with some markers that meet all these criteria. One is interleukin-6, satisfies all these criteria, robustly predicts mortality in a variety of settings. Muscle strength is one. The weaker people are, the less likely they are to live, and certainly we know strength goes down with age. Gait speed, FEV1, forced expiratory volume in 1 second, pulmonary measure. Peak VO2 max also. Let me show you some data from the Health ABC. We didn't do peak VO2 max in Health ABC. There's a lot of cardiovascular data showing that it's associated with mortality. So these are gender-specific septiles, as it turns out, sevenths of various of the representations of IL-6, serum IL-6, grip strength, walking speed, and FEV1. And you can see a fairly steep monotonic relationship. IL-6 is really remarkable, 8-year mortality, about 12% with the people at the lowest levels, no matter where you are, up to almost 40% mortality at the very highest levels. So, and it doesn't matter, this has been seen in lots of data sets, doesn't matter what the underlying condition people have, it still predicts. So does strength, so does speed. FEV1 isn't done as often, so I can't warrant to you that it's as useful or works in every setting, but in many, many large cardiovascular trials, it's a very potent predictor of mortality. So I'm not the only one in this game. You know, there is an industry in aging research of people coming up with their own markers, so I just don't want to say that that is the be and all and end all. And my colleagues, Ann Newman and Jason Sanders, also analyzed health ABC data and have come up with a death and disability index by something called a modified physiologic index score. They're starting from a different set of premises about subclinical disease rather than the set of premises that I used. And they have a score based on blood pressure instead of lung forced vital capacity, a different lung measure, forced vital capacity, which is how much air you can get out of your lungs, a cognitive test, the digit symbol substitution test, a marker of kidney function, cystatin C, and cut points of fasting glucose based on ADIA criteria. And this, when you make a scale of it, you get a very nice Gaussian distribution in the health, aging, and body composition population, a very striking death rate gradient from very small to very high. Of course, there are hardly anyone this good and hardly anyone this bad, but and then also ability gradient to predict disability. So there are lots of ways to put data together, either on their own or as scales or as you know, confected measures to get a handle on who's likely to die and who's not. And you might think that if these change in a positive direction with an intervention, there's a good bet that people who experience that change will live longer. So I wanted to look at this collection of measures in randomized clinical trials of older adults that had weight loss as one of the components. And these are all the ones I could find in the literature. And you got on this list by two criteria. Criteria one is the average age of the population was 65 or older. And so that cuts down the sample size tremendously. And the other is the design had to allow you to isolate the effects of caloric restriction. So in every case, it might be weight loss and exercise. Let me put it might have a weight loss and exercise group and an exercise group. So you could get an idea what weight loss was doing by subtraction. For example, and that's this case. So con means control, exercise plus caloric restriction. You can figure out what caloric restriction is doing by taking the difference of these two groups. Some cases, like this one, is a 2x2 factorial design. So you have people either getting exercise plus caloric restriction on its own, exercise is on control. So somehow you're able to isolate the effect. It's not, the effect of caloric restriction isn't completely confounded with some other factor in the design. So you can see these last anywhere from 9 months to 30 months. Quite a variety of weight loss achieved from 4% basically up to 10% weight loss. A variety of background things that to Messier, Kritchevsky, and these three are weight studies. This is hypertension. These are just people who are frail and obese. And this are the range of biomarkers from those previous two lists that have been assessed in these studies. So there's some work to do to fill out this table to see what all the potential functional benefits of weight loss may or may not be in older people. Lots of work in gait speed and IL-6 and some others. So what do we find? So let's first talk about gait speed. There's a great paper. If you're not familiar with it, I encourage you to look at it because it's sort of remarkable. It's a data pooling project looking at gait speed, predicting mortality, pooling about 8 or 9 different cohorts together, and find very consistent and robust finding that for every 0.1 meters per second difference in gait speed, there's a 12% difference in mortality. So gait speeds tend to run anywhere from about 0.4 to about 1.2, 1.3. So that gives you an idea. for example, if you're going to increase gait speed by 0.05 meters per second, you would expect that to confer a 6% mortality advantage in people who sped up. So I'm not going to go through all of these in great detail, but they're all set up the same way. You have a baseline gait speed, caloric restriction minus controls. So that gives you one way to look at the caloric restriction effect, caloric restriction and plus exercise minus the exercise group. And you basically get about between a 3 and 4% increase that translates into about 0.04 meters per second speeding up. And this is very consistent. It's shown in ADAPT study, Dennis Villarreal study in obese and frail older adults. They did a fast pace and got even a bigger effect. Jack Kritchevsky in a community dwelling group, same thing. In the IDEA study, a little harder exercise, a little bigger caloric restriction, also 0.5 meters per second. So for some reason, just losing weight speeds older people up. That might have to do with the fact that losing weight stimulates mitochondrial biogenesis and more energy to walk, or it could be just they have less weight to move around, and so they can go a little faster. Effect on strength. This is a big surprise, and this is a conundrum or a thing that needs to be solved in aging research. So what we see here, is looking at, this is one repetition max, 1 RM summed across several muscle groups. In the control group, doesn't change. Caloric restriction doesn't change. Exercise group, which included resistant exercise, goes way up, and caloric restriction and plus exercise goes way up. So as expected, losing weight on its own doesn't make you any stronger. But what they found looking at thigh muscle volume imaged by MRI is a 6 or 7% decrease in muscle volume with caloric restriction, yet strength didn't change at all. So in older people, there's something about what we image as muscle volume that is unconnected with strength, because we can have people lose muscle volume or muscle mass by DEXA, and we've seen this in several of our studies at Wake, yet seeing that they don't get any weaker. The participants don't get any weaker. So there's something going on, and people have coined the term, I guess, Todd Manini and Brian Clark, dynopenia, to reflect age-associated weakening as opposed to sarcopenia, which is age-related loss of muscle mass. And these ideas are sort of conflated over time. But there is something going on. So we can get older people to lose muscle mass, apparently, but they're not getting any weaker, which is a good thing, because we don't want older people to get weaker because of how important that is to their functional health. Interestingly, Dennis Villarreal did this very carefully. This Yassine study was just in a handful of folks. Saw caloric restriction on its own led to a 10% increase in VO2 max. And it adds to the exercise effect. I have a colleague, Delane Kitzman, who's basically repeated this study in people with heart failure with preserved ejection fraction, seeing exactly the same thing, and showing also in his data, which is yet to be published, that it's not just attributable to the way the equation works in dividing by body mass. There is real, actual increase in ability to utilize oxygen. Certainly IL-6 is famous in gerontology because of a lot of interest in this. There was a paper, 2007, IL-6, the cytokine for gerontologists, showing how it seems by giving this to animals, you seem to recreate a lot of the problems that many older adults have seen to have. These are data from Steve Messier's study, the ADAPT study. These were overweight people with NEOA, and comparing exercise and diet in combination. And you can see in the control group over the 18 months, not much change in their IL-6 levels. Exercise, a little bit of a decrease. This is not statistically significant. But both the diet and diet exercise, a substantial 11% reduction in circulating IL-6 levels. And two other studies have looked at this too, and they find even bigger changes. And everyone thinks that exercise is super anti-inflammatory. In older obese people, it's not. Losing weight is anti-inflammatory, but exercise on its own has a small effect, but not nearly to this level. So what about other biomarkers? Well, there's lots of data from younger groups and confirmed in these studies that you'll see a small reduction in blood pressure in older people. Lots of data from younger folks, and again, from these studies showing a reduction in plasma glucose. Really no data in lung function. Recent data on psychomotor speed, finding a better performance on Trails B, which is somewhat psychomotor speed, and executive function, but really very little data, and no data on kidney function. Though in the look-ahead trial, the people randomized to the intensive lifestyle intervention had a slower progression of kidney disease, so it may be helpful. We don't know in older folks if it would help. So this is a summary. So where there's data, weight loss does all the things you would hope caloric restriction would do based on the animal model. It's so far as the biomarkers have been assessed is concerned. So maybe this would be a good panel of measurements to use if you were trying to evaluate a life-extending treatment in older folks. So let's talk about if we can go the whole way. So we have epidemiologic data that leads showing these things predict mortality. We have clinical trial data that's showing that weight loss interventions positively affect these markers. Can we skip the middle and skip to the end? Well, maybe, and maybe not. So first, these are data from a study called the Swedish Obesity Study. This is not a trial. These are a longitudinal follow-up of obese folks in Sweden, it turns out, some of whom receive various kinds of gastric bypass surgery and some BMI matched controls. And these are showing the weight trajectories over going out to 15 years, showing substantial and sustained weight loss in the people having their stomachs either bypassed or restricted in some way. Compared to the control group. This slide is their mortality experience. So really up to the first 4 or 5 years, there really is no difference in the mortality experience between the two groups. But then the curves start to separate, and over the 10 years, they end up being a statistically significant benefit in those receiving the surgery. Now, this is observational data, and so there are lots of confounders and selection issues that can't really be completely and confidently addressed in this. I would point to one thing which is kind of interesting is the timing of when the curves begin to separate. You could imagine that you spend your whole entire adult life exposed to a certain physiologic milieu which winds up and leads to various compromises of organ systems and the way your body works together. If I do something overnight, that's not all going to unwind the next day. It's going to take some time for all those risk factor benefits to play out to show an eventual benefit in mortality. It's not going to happen overnight. And this is very helpful in gauging how long would you have to observe a group that you gave something to that you think had these pervasive metabolic until you began to even see differences in mortality. We typically in our aging epidemiologic studies throw away the first 2 years of mortality, figuring that whatever killed those people was already present in some substantial form. We maybe didn't recognize it when we assessed them. And there are all kinds of examples where results turn around or go away when you disregard the first 2 years of follow-up in epidemiologic studies. So another study I've become involved in, which is not older folks, and nor was that one, we can't get all the way there, is the look-ahead study. Some of you may be familiar with this. It's sort of a, just an incredibly long, incredible study. The idea of the study was to see in people with type 2 diabetes, did an intensive lifestyle intervention, which primarily consisted of weight loss, but also a piece of that was some more exercise. Would that prevent the onset of new cases of cardiovascular disease in those groups? And so they enrolled over 5,000 folks, and some went to a support group, and some had this intensive lifestyle intervention for 4 years and maintenance and counseling thereafter. And so this is a quick summary of the slide of the design of the study. This is from the 2013 paper. This study was ended for futility. They got to the point where they said, no matter how long we go on, you're not going to show that this prevents cardiovascular disease. Doesn't mean it didn't show anything, but for the primary endpoint, they decided they weren't hurting anybody, but it was clear that in the way things were going, the main endpoint goal was not going to be reached. However, what they did achieve is really quite remarkable. First of all, a very large weight loss effect of a 7% weight loss differential in the first year, and then obviously some rebound on that, but we're able to maintain a differential of about 4% between the intervention and control group, even through 10 years. Big difference in physical fitness, again, rebounded after 4 years. Waist circumference also maintained a difference. Glycated hemoglobin, a reduction, and being able, so obviously through time, not all the impact of the first year or first few years, but able to maintain a delta between the groups for a substantial, up to 10 or 11 years. And they saw a lot of things in there that would, using this sort of analysis that we've gone through on biomarkers or risk factors, would expect to improve mortality rate in this cohort. Fitness improved, systolic blood pressure went down, renal function is preserved, depression is lower. It turns out that depression in the context of diabetes is a strong mortality risk factor. Gait speed improved, hemoglobin A1C. So based on observational data, you would expect this would have a positive impact on mortality. This is just showing that look-ahead is published on all of these endpoints, showing a benefit for the look-ahead intervention. You could guess at what might happen with mortality. So they were able to show a 0.6 MET difference between the two groups at year 4. And if you look at the equations, this would translate into a 15% mortality advantage for the people in the lifetime intervention over time, if that's the way it worked. So this is what is found for mortality. These are the published data. In 2013, they found a hazard rate ratio of 0.86, so a 14% reduction in all-cause mortality. It was not statistically significant. These are the survival curves between the two groups, with the intensive group, the weight loss group, after about 4 years, beginning to show some mortality advantage over the DSC group, the usual care group. So it's interesting how this lines up almost identical divergence with the Swedish obesity study, where they had 15, 20, and 25% reduction in body weight. This is only 7% and only about a 4% delta sustained. So it's more than just how much fat you have. There's something else going on here. I understand that these curves are continuing to diverge, but I don't know the data and I can't share much with you. So this led us to do a meta-analysis of all-cause mortality in trials of weight loss, comparing the weight loss arms to the non-weight loss arms. 2 of these contributing papers are by our very own Dr. Shea, or your very own now, Dr. Shea. And there aren't many out there. I mean, there are a lot of weight loss studies. Most of the time, it's weight loss in both groups, but one group gets a drug to make it work even better. There are probably 1,000 of those articles. The ones that have weight loss on one side of the balance and not on the other are relatively few. And the number that also report mortality endpoints is very small. So we looked, Kristen Beaver is a postdoc working with me. She and I reviewed 3,000 abstracts, 4,000 abstracts, ended up with this 13 or 14 studies here in there. They look, you know, they don't, many of these are fairly short term, not a lot of events, so there's very wide confidence intervals. But when you do the requisite things, you end up with a 0.85, a 15% reduction in all-cause mortality. That certainly excludes the null value, or, you know, just sort of kisses a 1. There's a lot of concern in older adults that losing weight will kill older adults. It's certainly true from geriatricians. If you talk to them, they don't, very reluctant to ask older people to lose weight. This gives you some security that at least you'll do no harm, but there's some evidence that it will actually lead to a longer lifespan. These are under review. We had a very easy review at PLOS One, and so we'll expect this to come out fairly soon, but I can't guarantee it. So summary, weight loss trials provide, I think, a first approximation of effect sizes and potentially responsive biomarkers to evaluate caloric restriction mimetics. So this is a translational goal. And it could be many more than the ones I've presented here. I'm sort of providing a framework. I can't tell you, and probably it'd be better to use composite biomarkers than individual biomarkers, but I think a lot of that has to do with how you think the thing you're giving, how it acts. Can I tell you this applies to non-obese adults? I can't. All of these studies that I show you are all done in overweight and obese adults. And so this may be the benefit of treating obesity. It may have little to do with life extension because of slowing aging. The extent which some of the benefits are due to reductions in mass per se isn't clear, and this is particularly concerning with gait speed. So if I took you and put a vest on you that added 4% to your body weight and asked you to walk back and forth, I bet you'd slow down, and I bet if I took it off you, you'd speed up. Some of the more, the IL-6 I don't think that pertains to, but might. And it's also important to note that all the biomarkers I showed you, these are important outcomes in and of themselves, even if this drug didn't extend lifespan. If you had a drug that made people stronger or sped them up or reduced their blood pressure or slowed the decline of kidney, you're doing a good thing for people. So quite independently of their value as a way to evaluate the value of longevity treatments, if they did any of these things on their own, they should be very valuable to an older population. So I'd like to acknowledge the support of our Pepper Center. This is our one to do stuff in the Look Ahead trial, and collaborators who've worked with me on a lot of thinking through a lot of this stuff. And that is it. Thank you.
Thank you very much for a very interesting talk. The floor is open for questions. Alan.
So I was wondering if you could go back a little bit to your distinction between intervention, short-term intervention, and then the time when you should look for the response to the intervention. And specifically, maybe reflect about that Musak's paper, where they did like a 5-week glycemic index intervention, and they showed that had no real effects on cholesterol or whatever. But I wonder if, so really what I'm wondering about is, they look for short-term readouts, but there could be very useful, or maybe not, a long-term, actually salutary effects of such a diet that could be missed.
Right. Right. So I think as far as, let's see if I can get this to, It's sort of like a dream in all flesh. So the short-term stuff I see as adding reserve. The short-term benefits of exercise in an older person is adding reserve. We know, for example, that master athletes who are elite aging lose muscle mass about the same rate as non-athletes do, but they have a lot more reserve. You can build reserve in the short term. If you can sustain it, it will help you stave off the length of time. Now, what I think you're talking to is also an important feature and a slightly different but related question, which is we can do metabolic changes and we can afford to see what the short-term effects are. But if we're right about the underlying biology, they're also going to have long-term benefits, but those long-term benefits often get diluted with other physiologic things that are going on in people that sort of sum to health, longevity, or earlier delayed mortality. So there's a lot more variability. So if I'm doing a short-term study, I can get people to do that thing I can get almost anyone to do eat anything for about 5 months or 6 months, but 18 months, you know, the sustainability of these things to show a long-term benefit means the sample sizes have to go up to account for that, and there are all kinds of variability in the other kinds of things that will also influence the long-term outcomes I'm trying to affect, which also add to the variability which makes my sample sizes go up. So I think it would be wonderful. I mean, that's why the look ahead is such a treasure, is because they went and sustained something for 11 years. I mean, you know, studies have done that. They're like 2, this one and Women's Health Initiative, and that's about it. So it's really, these are very valuable for trying to understand how these things unwind over time. So I think conceptually I'm right with you. These things ought to have a long-term benefit. Practically, it's tough to get anyone to pay for it. So, sir.
At the very end of the talk, you mentioned, you posed the question whether we ought to continue looking at these individual markers as endpoints,
right,
or perhaps whether the primary endpoint ought to be an integrated composite index.
Right.
So to what extent have people created these composite indices, and when that's been done, do we learn anything additional by looking at a composite index as opposed to looking at individual markers? I mean,
right,
just to play devil's advocate,
yeah,
I could argue that you might obscure certain things, right, by trying to mix apples and oranges. Yeah,
no, I agree. I think I pose it as a question because I don't think there's a single answer to that question for exactly the point you bring in. People have been making composite indexes of your biologic age. You can go on the internet and say, what's my age? And you can put in, do you smoke? And it'll tell you what your biologic age is. I think in order for things to qualify to be part of the composite, they have to show each individually a responsiveness to the change of the pathway that you're trying to affect. Otherwise, you're just adding noise. And if all of the things in your composite are responsive, I could see where combining them in some fashion might dampen the noise that might be associated with any individual one. But if that weren't true, and you didn't already know that all the things worked in the way you'd hope they would, then I think you're shooting yourself in the foot by making a composite. In the back.
Thank you for a very nice review of this area. I have a question about the sort of the nadir of the BMI mortality curve.
Sure.
For the population. So since that nadir, at least in some trials, appears to be in the overweight range, if weight loss continues below that nadir, does that have an influence on some of these biomarkers? Do they actually revert and
I don't know of any data that speak to that question. I think all the trials of weight loss are in people who are overweight. Some people could be in these studies with BMIs of 27, which is about at the nadir, and we have people in those trials, and we've never gone to look to see, maybe we'll go back and do that, if people who get pushed well below that show a benefit. I think there's a problem, a conceptual problem and a confusion is that the BMI-UR is an integrative measure of your cumulative energy balance over your entire life up to that point. And so BMI at a given day doesn't really tell you that much. If I have a BMI of 35, it's because I've been in positive energy balance most of my adult life, and I'm probably heading up. If my BMI is 18, it means I've probably been in negative energy balance at least for the past 5 or 6 years, and I'm probably heading down. So there's a confounding between the underlying physiologic process that got you to the BMI that you are and the health outcomes that you're having. And people have been interpreting a single slice BMI as telling you, as being the indicator. It isn't. It's a proxy of an integrated physiologic process over time, which is what you'd rather have measured. So people, and I haven't seen this paper myself, and maybe people in the audience are familiar with it, that came out relatively recently. Well, people looked at peak weight, not at BMI based on peak adult weight, not on the day it was measured, and found quite a different relationship with mortality, for example, than on the day that it was measured. So I've tried to be very cautious in interpreting these cross-sectional snapshots of physiology that are really much more complicated.
Steve, this is perhaps another loss on the question of sort of indexes,
yeah,
or clusters of biomarkers or observations. If, as we know, every cell in the body has a clock, and if it's certainly possible that not only different cells will age independently from one another, but organ systems and their functions will age independently to some extent from one another, how much do we have to give care to extrapolating to aging, the larger issue, from observations in one organ system or one function or one set of cells? Isn't it possible that what is really going on here are agings, just as there are cancers and not cancer?
Well, I think that's a very important point. I think data from the CHS All-Star study has looked at the correlations between aging of systems over time, and to your point, find relatively small correlations to the way the cardiovascular system aged versus other organ systems. And I'm sort of conflicted because I get that point and I'm very sympathetic to it. On the other hand, there's no doubt that caloric restriction makes animals live 40% longer, and it affects many organ systems. And so we have this sort of, and so I'm not exactly sure in my own mind how to reconcile those. I do think there are certain things that seem to affect the milieu that may promote aging across a variety of organ systems. Caloric excess seems to be one of those, and NF-kappa B activation, and things that damage tissues, causing the clocks to run faster, may affect a variety of tissues. So I think there are sets of tissues that may work together, and I don't believe there is a that we're all on one big clock. I think you're right that different organ, it's sort of time to whichever organ system fails first is what's going to get you. And so, so I'm fumbling around because I don't have a great, I haven't resolved in my own head how to resolve what my starting point was very much along the lines that you expressed, and the animal work showing that you can make animals live longer with giving them these things. So.
Just a simple little question.
Yeah.
What role do you anticipate that the individual's genome is going to play in this role, in this process of predicting longevity and how to mitigate it at the individual level?
Okay, so here's my flip answer, between 35 and 65%. Because every genomic study I've ever seen, that is the genomic contribution is between 35 and 65%. When people have tried to use long-lived populations to identify genomic targets, and with the exception of ApoE and one of the FOXOs, have not really been very successful at it. It seems that extreme longevity does appear to have a genetic component, as well as extreme early mortality also has a genetic component, but really between about 55 and 99 or 95, it's been hard to pin down specific genetic contributors. But I think obviously, depending on how things are behaving and what genotypes you have in these pathways, some people are going to benefit from these interventions and some are not. And I don't know that we know enough to predict which one is going to be helpful. I think the way forward is to try interventions and do genotyping and based on some hypotheses and see who is responding and who's not. To do those effectively, it's an order of magnitude bigger studies than people are used to doing. So that's another, there are practical issues having to do with scale that are important in trying to work those kinds of things out. But I know there's some genetic contribution. How to work through that is tough. Yeah.
Well, thank you very much.
Okay.